Okay, we're officially recording. RNA, I was always taught, was transcribed from the antisense DNA strand. And the reason for that being that then the RNA copy being transcribed, of course, you guys all know that transcription only happens in the 5' to 3' direction. So then your mRNA or RNA your RNA strand would then be in the same direction as your sent strand of your DNA. However, we now know that it also can work in the opposite direction. So I got to play with my whiteboard. I love whiteboards. I'm ready to fill it out for you. So I hope you guys can see this. Can anyone not see this? Okay. So in addition to going to using the antisense strand as a template for the RNA, it can also use the sent strand, in which case if it uses the sent strand, you're still going to be transcribing in the 5' to 3' direction, but now your RNA molecule is going to be in the antisense direction. So that would be considered antisense RNA. And then I'm going to kind of hop back and forth between my whiteboard and these kind of things that I found on the internet. And so basically what it looks like is you can have transcription happening in two directions. So this is just another depiction just showing both of what I just showed you. So here you have your DNA strand. Again, you have your sent strand on top. You have our antisense strand on the bottom. And when you transcribed using the antisense DNA strand as your template, you get your mRNA in the 5' to 3' direction. mRNA in this example, of course, that could be any RNA, not just mRNA. However, if you use your sent strand as the template, like shown here on the right, you'll see that your final RNA molecule that's transcribed is actually in the 3' to 5' direction. And so that would be considered antisense RNA, and that's actually one of the mechanisms by which RNA is able to self-regulate, which is pretty cool. But that also means in addition to that, we can also have overlapping genes. And that was what was one of the things that was shown in the paper that I sent. So what happens in biology is that genes are not just in unique locations. Genes are not transcribed from just unique locations on the genome. So in addition to one region, one location on the DNA being transcribed to make an RNA, there can also be regions within that same region or overlapping regions that'll transcribe a different RNA. And so in this paper that I put on that Google Drive, you'll see kind of a breakdown of overlapping transcripts that come from the same strand. And then also, as I just showed you, we can have sense and antisense RNA. And so that sense and antisense RNA can also come from overlapping regions on the DNA. And so I just kind of have a little picture here of what that looks like. So this would be overlapping on the same strand. So if you look here at the bottom, so we have the template here in black. So in black is the DNA template. And then these are three separate transcripts that I labeled as transcripts A, B, and C. And so as you can see, they're their own transcripts. However, they're transcribed from overlapping regions relative to the DNA template. So you'll notice that transcript A has this overlapping chunk with transcript B, and also this overlapping small chunk here with transcript C. And then transcript B is, of course, overlapping with transcript A and transcript C, and then transcript C is also overlapping with both of them. So that's what's meant by overlapping transcripts that are derived from the same strand, being, in this case, our antisense template strand to make sense transcripts. In addition to that, we can also make we can also make overlapping transcripts from opposite strands. So as you can see again in black, you have the template DNA. So on the top you have the sense template DNA, and on the bottom you have the antisense template DNA. And you again have two transcripts being transcribed here, transcript X and transcript Y. And so transcript X is coming from the sense strand to make an antisense RNA. And in pink, the antisense template is being used to make a sense transcript that I labeled as transcript Y. And you'll notice that there's regions of overlap right between the two. So that's what's considered by overlapping transcripts that are on opposite strands. And so when this becomes a problem is when we want to go ahead and make our libraries. And so let's say in the example I'm going to use here, we have that in our sample. Can you guys all see this?
Yep.
Awesome. Sorry, this is why in person is better. So as you can see in orange here, so disregard this top part now that we just went over it. So let's say we have a sample, and in our sample, transcript X, so the orange transcript here is the one that's expressed, okay? So now what we're gonna do is we're gonna take, during our library preparation, we have transcript X being expressed in our sample, and we do a chopping mechanism, right, to get these short. So let's say that I chop it right here, right, so that I'm left with this part here of transcript X, right? And you'll notice that this part here is overlapping entirely with transcript Y, right? But we know, because I told you, that it actually came from transcript X. And so what's going to happen is during your first strand cDNA synthesis that we just went over yesterday, we'll make a copy, right, of our insert of our chopped up RNA here. And then during second strand cDNA synthesis, so after this first strand synthesis, remember that the original template is removed. And so we're left just with this one. But we know that this strand should be the opposite direction, right, of where it came from, because of the nature of how they're transcribed during PCR. And so we have our first strand, cDNA. And now when we go to make our second strand, cDNA, this time, if let's say that this wasn't a stranded library, so that means that when we make our second strand DNA, it's made with DTTP instead of DUTP, like what I showed yesterday. So that means that we still have T's in both copies. So now when we go to do that final PCR step, the polymerase that we're using, because it can read through T's, is able to transcribe both in both directions. And so now what we're left with when we go to do our sequencing is we're left with both directions of our original template. And so now when we have this molecule as our sequence, right, and then we're going to map it back to the genome to figure out where it came from, well, it matches up exactly with this. Excuse my silly line. And it matches up exactly with this. And so if we don't know what the origin was of our template, we won't know which transcript we should assign that read to. And that's kind of the caveat of not having a stranded library, is you can have inaccurate counts of your transcripts because we have about, as this paper went over in the reference genome, we have about 19% of genes that overlap with one or more genes on the opposite strand. So there's almost 20% of genes in, this was a human reference, and we're very similar to mice. And so 20% of our genes can have overlapping transcripts on opposite strands. So if we don't maintain and know which strand our template came from, we have no idea what to assign a read to if it covers that overlapping region. Does that make sense for why we do stranded libraries and why they're important? Yes, no, no, yes. Still confused? Thank you, Felix. I do love my whiteboards. I have them all over my house. Does anyone else need any further explanation on strandedness and why strandedness is important for generating accurate counts of the transcripts? Okay, awesome. So let's get back to the lecture. And we can start with that FastQC, the duplication levels of FastQC. Before we get started, though, I did want to open it up. Is there anything else from the material that I covered yesterday that you guys want to hear again? Everyone's good with the material from yesterday? No questions from Felix. Awesome. How's everyone else doing?
No, that was a no. I do have questions.
Oh.
Sorry,
No, go ahead. Ask away.
difficult. So if you go back to slide, oh shoot, where are the slide numbers? Um, the slide with, oh wait, several slides have library preparation in trying to figure out the slide numbers.
You can just tell me what you see on the slide. I should be able to find it.
Okay, it's library preparation, and it is the second slide with library preparation in the title.
This one here?
Uh, let's see. Yes. What was RD1 SP and RD2 SP again?
Oh, so those are the priming regions. So remember, this kind of takes advantage of PCR. And so if you remember in PCR, so in PCR, we need primers and in order to amplify the region that we're interested in amplifying. So in the context of sequencing by synthesis, we have absolutely no idea what the target is that we want to sequence by synthesis, right, using the concept of PCR here. And so we don't know what that sequence is, and nor do we know what's going to be flanking it, because that's the whole point of doing it, right, is to sequence. But we need some way to prime to be able to get the polymerase to latch on and start transcribing, to start creating what's ultimately going to be our sequenced strand. And so in order to have a region that we know, so that way we can do that priming to enable polymerase to do its job, we add them on artificially. And so that's what's going on here. So this region here is going to be the region that our primers are going to anneal to. So that's why this is the first thing attached to either side of our insert, right? So we don't know the sequence of our insert. That's what we want to find out. And so we do know the sequence, because we're adding this on artificially, of this priming region and RD2 on the opposite strand. And so when those are attached on here, now the primers that we use during the sequencing cycle are primers that are complementary to these things that we artificially added on. So it'll latch on and we'll be able to start sequencing our insert of interest. Does that make sense?
Yeah, got it. Thanks. And then could you explain the side again, please? The 1 index, 2 index, and UMI?
Yeah, for sure. Okay. So do you understand the latching on of the adapters to our region of insert? Is everyone good with that? And we are going to start with that duplication slide. So these adapters that we add on, in addition to having that priming region, so that way we're able to allow the polymerase to hop on to actually sequence a region of interest. If you guys, in addition to that, we also need a sequence or oligonucleotides, ATCs and Gs basically, that are complementary to the flow cell. And so that's what the P5 and P7 are. So those are the parts that are going to be complementary to the little oligos that are on the flow cell that's going to allow the hybridization of our templates to the flow cell, which will ultimately allow us to copy it so that way it's tethered to the flow cell like we saw in that Illumina video. In addition to that, we also need to be able to, since we're combining sequences from several different samples, because as you guys saw in the Illumina video, we sequence billions of molecules at a time, right? So way more than what you need for just one sample. So what we do is we combine a bunch of samples together to sequence it all at once. And so if we do that, we need to be able to, once we do the sequencing for each short read that we get back, we need to know which sample that came from, since we combined the libraries from all samples together. And in order to do that, during this library preparation step, which is done on each sample individually, we add a specific index, which again is just a string of ATCs and Gs. And I'll show you that again on the read. And so this string of ATCs and Gs is going to be unique. So we have a unique set of ATCs and Gs for each sample. So that way we know that if in the read 2, so if you remember the Illumina sequencing video, we first sequence the forward read, and then we sequence this index here on the P7 side. And so that second read technically that we generate is this index here. And that set of ATCs and Gs, we allocated which sample that belongs to. So we know we have that information. And so we can tell the program, hey, if you read this set of ATCs and Gs in that second read of the Illumina sequencing, make sure that you tell me that this sequence goes to that sample that we put that specific index on, that specific set of ATCs and Gs. So that's if you have it just on one side. And so we do paired end sequencing. As I mentioned, some people would just do single end sequencing, in which case you're only going to have it in this direction. But if you do paired end sequencing, it's really beneficial to also have an index not only on your associated with your forward strand, you also want an index associated with your reverse strand. And so that would be on the P5 side of things. And so this is just another way of viewing kind of these adapters. And so what you're seeing here, these adapters represent single index. So you have your P5 and your SP priming region associated with the P5 side. You have your region of interest. You have your SP priming region associated with the P7 side. But then you also have this index on the P7 side. So that's what's being represented here. But for paired end sequencing, and to be extra confident that you're really assigning those reads to the correct sample, it helps to have an additional tag, right? So now if we're matching two tags, we're like, okay, we're definitely sure that this one is going with the sample that we put these two tags on. And so what happens in unique dual index is, in addition to having a tag on the P7 side, there's also a tag on the P5 side. So what that would look like in this picture is you would have another tag in a different color here. And so now when you sequence your read from the P7, which is what we do first, right? And then we sequence the read from the P5 direction. So if we have unique dual indexes, we'll have that index read associated with the forward read. And then when we do our reverse sequencing, we'll also sequence another index. And so now we'll have two tags for each sample. So that way it increases our confidence that we're going to assign that read back to the sample that it came from. Any questions about single versus unique indexes, or single versus dual unique indexes, before we go on to the UMI. Okay, nay. Is it too early for everyone? Thumbs up. Okay, awesome. And then in addition to having these indexes, and this we're going to get into, so this UMI will probably make a little more sense in the slide after the last one we went over yesterday. And so what can happen is, remember, the next step of this is to do a Pcr amplification. And then that's going to make multiple copies. We're going to do like a low cycle, but if there's a ton of a particular gene that's being expressed, when we do this Pcr step, we're going to increase those copies even more. So that way, when we combine them all together, and then we go ahead from all the samples, combine all the libraries together from all samples, and then we go to sequence it. Remember, we have like a solution that contains like millions, if not billions, of these short sequences, right? So we have billions upon billions of these short sequences that are associated with the different samples from our experiment. And those short sequences come from this library. And because we do that Pcr step, if we have a gene that was really highly expressed in a sample, during that Pcr step, we make it even more expressed, right? Because we're making copies of it. And so when we go and take our solution and load it onto our flow cell, the likelihood that we might get not only the original part of that gene, we could also get a copy of that gene that we made artificially during this Pcr step. And so, of course, when we go to do our quantitation now, we added something on artificially that didn't come from the original biology. So it didn't come from the original sample. And so we want to be able to account for that. And so one way to do that is when you're making your libraries before you do that Pcr step, you add an extra flag here, this UMI, and again, just a set of A, T, Cs, and Gs. And the longer that is, the more unique it could be. And this is random. And so this is kind of a random set of UMIs. So we don't know which UMI belongs to what, right? Because we want this UMI to be unique to each molecule. So unique to each molecule within the same sample and unique to each molecule within different samples, or unique enough to reduce the likelihood that the same flag is going to be latched on to the same insert. And so by doing this, by adding on this flag here, and we do it before the Pcr step, now when we do that Pcr step, if we make a copy of this, and then when we sequence, in that sequence, if we see the same exact insert sequence, and we also see the same UMI sequence, well, we know we added that UMI on artificially before Pcr. So if we see a copy of that UMI along with the same sequence in the insert, then we know that we added that artificially. And so we probably shouldn't count it when we do our quantification of our the number of reads. Does that help at all? Does that help? Does it make things more confusing?
That
I'm
helps.
sorry. I had a question about that.
Go.
If you have a UMI and you want to see if you have like multiple UMIs and you're like, oh, okay, this one's the PCR'd one. How do you know which one's the PCR'd one and which one's the original? Because you're changing the original now?
So great question. So that's why we add this on during this library prep before PCR, right? So before this step of the library preparation, the only thing we're sequencing is our insert, right? Which is the sequence that we're curious about, that we want to know about. And then we have the indexes that we're going to be sequencing, right? And we know those sequence and we know that they're associated with certain samples. But then each sample will contain multiple reads, right? So if you remember the read depth, we have like 40 to 60 million reads is our goal per sample, right? So 40 to 60 million reads per sample, which means there are 40 to 60 million reads that all have the same indices to know that they belong to the same sample. But within those 40 to 60 million reads, we want to know if we made any artificial copies during that PCR step. And so in order to know if we did, during the library preparation, we add on this random set of A, T, C's, and G's. And so now when we have our reads back, right, in addition to our sequence of interest, so now if we have a sequence of interest in our read, and if we have another one that's the same set of A, T, C's, and G's, well, it could have come from biology, right? Because we could have just multiple copies of a transcript. And like I said, even though that chopping up of the transcripts is supposed to be random, there's always going to be some bias associated with it. So it can happen to where if you have a really high expressing transcript that you have multiple copies of it. So when we chop it up in the previous steps, we actually have the same exact sequence, right? And so then when we go and get our reads back, we see two reads that are identical sequences. That could have come from biology in the way that I just said, right? But it also could have come from the PCR step, right? It could have come from this PCR step where we're making copies artificially. And so that's why we add on just before we do the PCR step. So right now, before this step, the sequence that we're interested in came from biology, right? It came from our samples. That's what we're looking at. We want to know how much of the transcripts are there. So in order to ensure that if we happen to have the same exact sequence from biology, we want to make sure we can identify when it comes from biology versus when it comes from the next step where we start making artificial copies of our transcript. And so before we start making those artificial copies, we add a UMI, which again, set of A, T, C's, and G's. They're going to be unique. Let's say unique for our purposes, not entirely unique, but let's say unique for each read coming from that sample, right? So we have, let's say 60 million reads coming from one of our samples. And of those 60 million reads, they all have the same index, right? Because they're all from the same sample. However, each read is also going to have a unique UMI. But now, if we have a read that is the same sequence of interest, right? So we have an identical copy of a read, so it has literally the same sequence. We look at the sequence of the UMI. So if they all start off with unique UMIs before the PCR step, the only way that a read can have a non-unique UMI, which means a duplicated UMI, is if it happened during the PCR step. Does that help at all? That makes a lot of sense. Oh, awesome. Great. Good. I'm glad because some of you guys are going to be helping me figure out a pipeline to be able to pull out these UMIs and remove those duplicates that come from PCR. Awesome.
So I have a question.
Yeah.
So when you look at sequences, I assume that you get a lot of those duplicate UMIs in the samples. Does it definitely correlate with the highly expressed genes, that you get a lot more of those with, that you would assume that if something is highly expressed, that you might, it's really critical to have the unique one so that you can tell whether something is genuinely highly expressed or just relatively highly expressed and then amplified, right? So with each amplification cycle, which I remember that is limited, the number of cycles.
Yeah, we do about 15. Is Marie on? You guys still do 15?
Yes, we do 15. So for that, there is a limitation, yes, you did, because eventually this is called linear amplification. So what you're doing is you're doubling your concentration every cycle you do. And so at some point, it saturates, right? And your information just becomes more copies. So.
Yeah, so the goal is to get it somewhere in this linear phase of the cycle. So that's why they do about 15 cycles, because you'll see at 15 cycles just in this example that most is in the linear phase. But at that point, you've already made copies of transcripts. And so that's where, especially if you have more, so now this is all like in a solution. And so the higher abundance transcripts, which you've now already made some additional copies of. You've also already made additional copies of some of the lower expressing transcripts too, as well as the moderate expressing transcripts, right? So in the goal of this PCR step is to be unbiased in the libraries that, in the reads, I guess I should say, that you amplify. So it should be unbiased. However, if you just start with more material, you're going to end up making more copies of it. So if you have a higher expressing gene, it starts with more material already for that gene. And so when you make copies of it, obviously you're going to increase the copies of the higher expressing ones sooner. And so after that 15 cycles, you'll have, more of your higher expressing ones, but you'll also have amplified your lower expressing ones, which is the goal of the PCR, is to be able to actually capture those lower expressing ones. But then this is all in a solution, right? So then when you go to pipette it into your flow cell, those are latching on randomly on the flow cell, again with some bias to smaller inserts, to smaller libraries. And so as that's randomly latching on, the likelihood that you're going to get a PCR duplicate from a higher expressing gene is going to be higher than the likelihood that you're going to get a duplicate from a lower expressing gene. However, all of those reads are going to contain a UMI. So even if you get the same sequence and the same UMI duplicated from the higher expressing one and you want to remove it because you know that you add that on artificially, the same would be true for a lower expressing or a moderate expressing one. It's just the likelihood that you'll pick up both, you'll pick up two copies that are identical from a lower expressing one is just lower.
So a follow-on question to that is, so I know when you load the flow cells, you don't overload the cells, you underload them so that not every well would have a chance of capturing an RNA or a copy. So have they done calculations of actually what is the efficiency of that step in terms of molecules that actually get captured?
Oh yeah. Illumina's done a ton of analysis and QC in terms of concentration of your libraries. So how much of your libraries you add onto your flow cell based on the size of your flow cell and what you should expect for the percent occupancy, because you're right, you don't want to overload your flow cell because then it makes distinguishing the colors more difficult. And so they're usually, what's the goal? The goal is usually what, like 70-80% occupancy?
Yes. So it's calculated so that basically the sweet spot, they call it, is around 70 to 75%. And at that kind of loading, you basically optimize the detection optically, right, on the flow cell. Because otherwise, when you overload or underload, you miss the depth. So you basically layer too much or layer not enough for the optical detection. So then you lose your depth. So there's like the standard depth that they go by, but then we try to overachieve it to basically maximize the depth that we can get.
Yeah. Does that answer your question, Sigrid? Do you have a follow-up?
Yeah. No, I'm just thinking about, very low copy number things and what the likelihood of them being captured is,
Yeah, and so that's kind of also been done too by inserting references like universal human references, the one that Illumina uses all the time. And by inserting like known concentrations. So that's another reason why we do the ERCC spike-ins. So the ERCC spike-ins, we know the concentration of those going in. And so let's say if, so it's kind of some of the data that Marie showed in an internal GeneLab meeting. Sorry guys, I know we're going a little bit in the weeds here. But so you can basically spike your samples with something with a known concentration. And then when you do your sequencing depth, if you picked up, so our ERCC is artificial genes that we make along with our libraries. And so those artificial genes, we know the concentration of them. And what we do is some of them have higher concentrations, some of them have lower concentrations. And so when we get our sequences back, when we look at our reads, we see for those ERCC genes, if we sequenced at, say, 60 million reads and we didn't pick up the lowest expressing of those transcripts, we know that in our samples of interest, we probably also didn't pick up transcripts that were at that low concentration. And so if you are really interested in low expressing transcripts, any guesses what you would do in terms of your sequencing? You guys shout at me, type it in the chat. So if you have really low expressing transcripts and you want to be able to detect them in your sequencing, which means there's very few of them relative to all the other transcripts, how do you think you'd be able to capture them?
Increase your read number?
Exactly. Yeah. So you increase your read number. So if it's 60 million reads, you're not capturing. So if, let's say you have a gene and you have an idea and you know that it's fairly low concentration, but it's a really important gene and you want to know it, and you do a run with an ERCC spike in with known concentration. And when you go and do your analysis, it's 60 million reads and you're not able to detect those low concentration of the known ERCC, you probably also didn't detect your low concentration of your gene of interest. And so you would want to sequence even deeper. So then try 100 million reads and see if after sequencing 100 million reads, if you capture those lower expressing transcripts. Any other questions about library prep, tagging, indices, UMIs? I know it's a lot of very technical stuff, but as bioinformaticians, it's important to know these things, to try to understand how to give your customer what they need.
Are you going to be covering the ERCC, the spike-ins in your lecture?
No. I think I already have so much information in here that, you know. Any other questions before we dive into that last slide that we ended with yesterday about any of the material that I covered?
Yes, slide 36 on your screen.
Sequence length?
Yes, so it looks like in the end you have 150 plus or minus one base. How is that possible?
Yeah, I don't know why it's graphed like that. That's not true. I was always curious about that too.
Okay, so it's not actually that.
Yeah, no, you're absolutely right. It really should just be one peak here. I've always been curious about that. They're all 150.
What? That's even more magical. How do you get what single nucleotide resolution? Because you start with a random distribution, which you saw in your electro micrograph. How do you end up with just one length? That sounds...
Because you control the number of cycles. So when you load your flow cell, so when you load all of your libraries, which you're absolutely right. So the libraries are different sizes. The inserts, as you mentioned, are different sizes. And of course, you know that they're different sizes because you did your due diligence and you ran your tape station and looked at your electropherograms. And so you see this range of sizes for your library, right? So you're right, there is a range of sizes. However, when you program the sequencer, you tell it how many base pairs to sequence. And those in Illumina's world are called cycles. So you tell it, I want you to sequence 150 base pairs. And so Illumina will only sequence 150 base pairs and then it'll stop. And it'll sequence it for every single strand that's on the flow cell.
Ah, I see.
So
Okay.
even though you have some reads, it'll have longer inserts. So that's why you really want to be good about generating your library sizes and doing your filtering, because you could end up, right, if you're sequencing 150 base pairs in either direction, right, so in this direction and this direction, if you have a 300 size insert, you'll be able to sequence the whole thing, right? You'll see it come end to end. However, if you have 350 base pair insert, you're only going to sequence to here on the forward read and to here on the reverse read, because you're still only sequencing 150 in this direction and 150 in this direction. So you'll miss that middle part of the insert that you had. And then another thing is that you could get overlap, right? So let's say that your insert is less than 300 base pairs. Let's say that it's 250 base pairs. Now you're going to get overlap. And so you're still going to have 150 base pairs on your forward read, 150 base pairs on your reverse read, but you'll see some overlap between the sequences of that forward and reverse read for that particular insert. And so you program the machine. You say, I want 150 base pairs every time. And so, right, it's like a computer. So if you tell a computer to do, it is a computer. If you tell it to do something and generate the same number every single time, you're going to get the same number every single time. Make sense?
Yeah. I guess then the only thing that I was wondering about was you mentioned that the flow cell is biased towards smaller, more shorter fragments.
Mm-hmm.
So how do you correct for that in your analysis, since even after the filtering, it looks like you have a range of lengths?
Yeah, so you don't really correct for it in your analysis. So when you go and do your alignment, it'll align based on the information that it has. And so the only way to correct for the bias of having smaller insert sizes is to be really good about your library prep. This is one of the reasons that I spent time to go over library preparation, because it's so important. So if we take all of the inserts here and run this on a flow cell, we're going to get a lot more of the we're going to sequence a lot more of the inserts on this lower end of the size of your libraries than on this higher end.
Yeah, so that's what I meant. Since you have that curve, then you know that distribution. So shouldn't you be able to use it at the end and say, okay, I see a lot of in that case, it looks like what, 300 to 1,000.
Yeah,
I see a lot of 300 and not so many 1,000, but I know that there's bias towards a 300, so I will subtract some of my 300 or weigh them less than the 1,000 because I know of this bias. Is there some way to do that since you know this curve?
Well, remember, you're randomly chopping this, right? So the representation of your transcripts, you could have, like for let's say you have multiple copies of one transcript. You could have sometimes when that transcript is cut and made larger, sometimes when that transcript is cut and made smaller, but that bias is going to exist for all transcripts, right? So it's kind of like when you make an error and like do +2 or something, but you do it to everything you have. Now, when you're comparing them one to one, that error is irrelevant, right? And so this bias of sequencing the shorter inserts versus the longer inserts, that same bias exists for every single transcript.
Well, is that true though? Because you start with, how long is a normal mRNA for a gene, I guess? If the gene itself was 200 base pairs or something. I don't know if that's feasible.
So now you're talking about correcting for transcript length. That you absolutely do have to do. So we do a correction for transcript length during the normalization part of our pipeline. So after we do our alignment and our counting, when we go through and generate, before we do differential gene expression, we actually normalize the data to account for differences in transcript length.
Yeah, I guess I'm thinking of a scenario where I don't know if this is biologically possible. One gene is 100 bases long, and another gene is 1,000 bases long.
Mm-hmm.
So before you sequence, you would shred both of them. So the 100 probably wouldn't get shredded, but then the 1,000 would get shredded into pieces that are 100 long.
Mm-hmm.
Or wouldn't the 100?
You're thinking about this perfectly. Like, yes, absolutely 100%. I promise we're going to address that exact question. That's going to come at the first part of tomorrow's lecture. So you're a little bit ahead, but I like where you're thinking, because yes, that is an issue. That is something we need to account for, because you're right. We could have, if you have a really long gene and that's chopped up, then now you have, even if you have one copy of a gene that's 1,000 base pairs, and let's say you chop it up into 100 base pair inserts, now when you go get reads for that gene, you're going to have 10 reads, right? 10 reads each at 100 base pairs to cover your 1,000 base pair transcript. Right now, if you have another gene that's also just expressed it with one copy, but it's only 100 base pairs, you're only going to have one read, right? And so if you were to just count the number of reads from each transcript, you have one read from your smaller transcript and you have 10 reads from your larger transcript, even though they only had one copy each originally. That's what you're getting at, right?
Yeah, kind of. Yeah.
So yeah, that's a problem, right? Because now we would say that, our longer transcript is expressed 10 times more than our smaller one, which is not the case at all. And so there's actually algorithms that are designed to account for that specifically. And so we're going to get into that tomorrow. So Yvonne, to answer your question, it's a little different than trimming. So we don't trim at that point. So this kind of comes after trimming, when we're taking a look at the reads and the number of reads, and then we just normalize those number of reads based on the transcript length using math. And we'll get into that quite a bit tomorrow, and then Lauren's going to get into it even more as she goes through her statistics lecture, and then the R portion of the analyzing our RNA-seq data. Any other questions before we hop back in to the lecture? They're all very good questions, guys. Important questions to ask for sure. Any other questions? Okay. Go ahead and get into it. Actually, before we do, let me make sure I know when I promised you guys a break. 10:40. Okay, cool. We have 40 minutes, and I'll adjust that schedule during the break. Okay, moving back into the lecture. Okay, so I'm just going to go over this slide again from yesterday. So we did talk about how to assess our sequences at the end of the day. I guess before this, just to kind of remind you all of what it is we are analyzing. So remember, we are analyzing our read. Our read, of course, looks like this. So there's 4 lines to every read. So this, during the FAST-QC, it basically takes this information of the sequence along with the quality scores that are given in association with the read. And it kind of compiles this information into a nice graphical format. So that's what we went over yesterday, kind of looked at the quality of the sequence at each base pair, overall for each read. We looked at the distribution of A, T, Cs, and Gs, and this equates to how confident we are in the signals that are generated. So we want roughly equal amounts of A, T, Cs, and Gs. We talked a little bit about the per sequence GC content, and that we expect it to be a normal distribution. And if it's outside of a normal distribution, it likely tells us that there's some form of contamination. We also talked about per base N content. So when the Illumina instrument is not confident that it can call an ATC or G, it'll give an N. And as you guys all pointed out, that happens very infrequently. Again, because there's just tens of thousands of the same strand that's being sequenced to really increase the confidence of the base calls. And then Felix, you made a good point here. These are all 150 base pairs. Exactly. I know this graph's a little weird. I never liked it either. We also talked about adapter content. So remember, if we have an insert that is smaller than the number of cycles we are sequencing, we could sequence into our adapter. And so basically, if our insert here, if we're sequencing at 150 base pairs and we have 100 base pair insert, like we just talked about, it's going to sequence 150 bases no matter what. And so it'll sequence your sequence of insert for the first 100, and then it'll go and it'll sequence into the adapter. And so that will show up, and the FastQC program is designed to detect common adapters. And so you guys all noted that we see universal adapters in some of our reads here. And so this last thing that we kind of ended on is looking at those duplication levels. And so again, this is the FastQC algorithm to do this. So there is some limitations to how it's identifying. So this is where it's actually looking at the sequence of interest. And it is looking at that sequence and it's looking to see, are there any other sequences? So are there any other reads that have the same exact sequence? So if there's 2 reads that have the same sequence, that's considered a duplicate, right? But we know that those duplicates could come from biology, but they could also come from some technical artifact like that Pcr amplification that we harped on this morning. And so another limitation of the FastQC analysis of duplications, it's only looking at the sequence of interest. So it doesn't look at UMIs. So it cannot tell if it came from biology, the duplication, or if it came from some artifact in the process of library preparation. And so there's that limitation. The other limitation is even if you have 150 base pair sequence, it's only looking at those first 50 base pairs. So that means if you have 2 sequences and they're the same in the first 50 base pairs but different afterwards, the FastQC analysis will still consider that a duplicate because it's only looking at those first 50 base pairs. Another caveat is this is giving percentages here, but it's not analyzing every single read in your sample. So it's only analyzing the first 200,000 reads. So as we talked about, our samples each could have, 40 to 60 million or even more reads per sample. So it's making these percentages based just on the first 200,000 reads. And the reason that it does this is because if it were to go and if the program were to analyze the full sequence length, if it were to analyze every single read in your sample, if it were to take into account UMIs, it would take a lot of computation to do that. So FastQC is really just trying to get you some idea of your duplication level in this very limited fashion. So that's why I always tell people to take the duplication levels in the FastQC analysis with a grain of salt. Because again, only looking at the first 200,000 reads, only looking at the first 50 base pairs of each read, and you're only looking at the sequence itself, not like UMIs or anything like that. So with that in mind, how you interpret this graph is the blue line here is going to indicate the percent of duplicated reads. Go ahead, Felix.
So when you say the first 50, does that exclude the adapter, or do you have to remove them?
So at this point, we're just looking at raw reads. So any reads that have an adapter in it, they're going to have an adapter in it. So that's a good point. However, if you recall from this slide, where are our adapters usually located?
At the end.
At the end, right. So one of the caveats, of course, of this is that it's only looking at the first 50 base pairs, right? So in this, it's only looking at like up to here. So there's going to be some reads where it will, detect adapters, and you're right? Because adapters are going to be the same every time. So I like where your head's at. So that could happen if we have kind of a really, really short insert or a primer dimer, or adapter dimer, I should say, where the sequence is only an adapter, then that would be a duplicate, right? So if we have sequences that are only adapters, we would see them starting off right here. And there might be some here, some kind of low-level ones that are only adapter dimers. And then those, of course, those adapters are the same sequence, with the exception of course, the index, which kind of comes towards the end. So yeah, so that could cause duplicates if it's in those first 50 base pairs. Does that answer your question? Awesome. Okay. I keep clicking. Here it is. Okay. So how you would read this is you take a look at this blue line, and your blue line indicates a percent of duplicated reads at a specific duplication level. Again, with the caveats, only first 50 base pairs, only looking at the first 200,000 reads. And so what you would see, like how I would interpret this blue line, is that we have roughly 20% that are unique, that have no duplicates, right? We have roughly what, like 8% where there's one duplicate. And then roughly 4%, again, of those first 200,000 reads that has three duplicates, right? So three sequences, or three reads with the same sequence. And it kind of goes like that, right, across. And then we have kind of these critical levels here. And so because RNA sequencing, we do have that PCR amplification step, it isn't uncommon to see some level of duplication, right? But usually if it's a region that you're interested in, like mRNA or pretty much mRNA, like mRNAs that you're really interested in, that you'll see a duplication level around here. So in that 1 to kind of 10 range. But then we can also see higher levels of duplications. And so here, what it's saying is that in this particular sample, about 10% of the reads have at least 10 of the same exact sequences. And then we see about 5% of the reads have 50 of the same exact sequences, about 10% of the reads have 100 of the same exact sequences, et cetera, right? So then it goes like that. And so why this is particularly concerning to me is especially over here on this higher end. So we have some reads, we have over 1,000 reads that are the same sequences, 10% of that, you know, and then over here, it's even more concerning, over 20% of the reads have 10,000 sequences or 10,000 reads that are the same. And so, as we mentioned before, these duplicates can be either biological or technical. And if your library is very diverse, you'll see basically this blue line here will be all the way at 100%. So you have a duplication level of 1. And then I mentioned this already, so it's common, specifically when working with enriched libraries, enriched being they go through that PCR step, to have some level of duplication. So I already answered that for you. And then as I mentioned, lower duplication levels like 2 to 9 copies are likely from interesting genomic regions like RNA or mRNA. And then moderate duplication levels, so around the 10 to 100 copy here, those could be derived from ribosomal RNA contamination, which we know is highly repetitive, and there could be a lot of it. So if you do have ribosomal RNA contamination in your sample, then it could come up here as duplicates. Or it could also be if there's a region in your transcript that's highly repetitive, then you'll also see it in the roughly 10 to 100 copies level. And then kind of these duplication levels that have over 100 copies of the same sequence, that could indicate a library issue. So either adapter dimers, like we were talking about, like Felix mentioned, or it could be too much Phi-X. So Phi-X is something, it's a viral genome that we actually spike as well into our libraries. And the reason we do that is to enhance the diversity in terms of the cycles. So we really want roughly the same ATCs and Gs in each cycle to help with detection purposes. And so Phi-X is used to help with that. But if you overdo your Phi-X, you usually spike in about 1% of the total library. And then we know that sequence, so we can remove it in silico. But remember, these are the raw data. So we didn't remove anything. We didn't do any filtering. And so you want to make sure, and your Phi-X doesn't have any indexes. So when you go to assign your reads based on your indexes, Phi-X is not going to be assigned. But it will show up as a sequence. So if that sequence is going to be identical, then we would see it in the duplication. And then we already answered this yesterday. Are there duplicates present in the sample, and should we be concerned? Yes and yes. Any other questions about duplication? We are going to be talking more about them in the next slide. But I'll just stop here. Okay, let's talk more about duplications. Oh, and then I have roughly what percent of reads are duplicates. So again, based on the first 200,000 reads, another thing this graph indicates is that if you were to remove the duplicates, which is a process called deduplication, you would be left with only about 26.5% of the reads, which means almost 75% of the reads are duplicates. Okay, so moving on to reasons for duplicates. So we talked a lot about having PCR duplicates, which is down here on the lower left. So we'll revisit that in a second, but I'm going to tell you some other ways that we can get duplicates, because PCR is not the only way that we can introduce artificial duplicates. So Illumina has two different flow cell types. It has one flow cell that's a lawn, and it doesn't have any like dimples or anything in it. So it just has kind of regions of the oligos. So the little clusters of oligos that are just evenly parsed on a lawn. And that's what you saw in that sequencing by synthesis video that we watched. So it's called an unpatterned flow cell. And that looks like this. So one issue you can have in an unpatterned flow cell is during that cluster amplification. If you have one cluster and it can if it takes on a weird shape, right, because it's kind of folding over super fast, making copies of itself. And so if you have it kind of fold over to where another region of the oligos are that are attached to the flow cell, it can kind of have this weird shape, right, and kind of almost look like two clusters. So that way when the computer is going to assess how many clusters you have or how many distinct reads you have, it could say that even though this came from the same insert, that it can count it as two. And so that would be, it would say, okay, these are two separate reads and they have the same exact sequence because in fact it came from the same original template. So that's one way in an unpatterned flow cell to get artificial duplicates. Another way is actually using the pattern flow cell, which is what we use. And so in the pattern flow cell, which you can see zoomed in here, we have these little dimples. And so the oligos that are attached to the flow cell are restricted to those little dimples. And so the idea is that this was supposed to help with this issue and that we have distinct locations of the oligos that are on the flow cell. And so the idea is that one insert would latch on here and make all the copies just within this little dimple. And so we would have basically more distinct circles to be able to call this, everything in here would be one read. However, what we see is that sometimes during the bridge amplification, we'll get a molecule that'll hop into an adjacent dimple. And so again, it would be the same molecule. And then during clonal amplification, if this is unoccupied by another transcript, it could hop in there and start clonally amplifying in an adjacent dimple. And so that, and again, each one of these little dimples on here are given back to us as a single read. So if that happens and we have the same molecule that occupies two of these dimples, they would be given back to us as two separate reads when in fact they came from the same molecule of interest. So those are two ways that these two types of flow cells can give us artificial duplicates. And these are very difficult to account for. And so another, actually we can account for these with the UMI because sequencing happens after we have UMIs. And then the other one that we've been talking about a lot is the duplicate that happens during the PCR step. So as we mentioned, during PCR amplification, we're making copies. And if it just so happens that our PCR copy One copy occupies one of those dimples, another copy occupies another dimple. That came from the same original molecule. And so now we have a duplicated read. And so actually all three of these we can account for if we have UMIs. Actually, this one could too. And so the next one is sister strands. And so what happens, as you guys know, our libraries, what's RTA? What does it say RTA? Oh, I don't know what that stands for. Single cluster that has been called as 2 by RTA. I think that's the name of the system that reads and interprets these individual clusters. I'm pretty sure. Does anyone know that answer? Lauren or Marie?
I don't off the top of my head.
Yeah, I'm pretty sure it's an acronym to describe the machine or the algorithm that's deciphering these clusters.
Googling flow cell RTA. I think it might be real-time analysis.
Oh, there we go. Yeah, Diksha beat you to it too. Awesome. Yes, real-time analysis. So during the analysis process. Thank you for looking that up. And then our fourth way that we can end up with duplicated reads is by sister strands. And so those are complementary strands of the same library. And remember that our libraries are double-stranded. And so just before the libraries are loaded onto the flow cell, they're denatured to create single strands. And so what can happen is that if we get a single strand, one of those sister strands occupies one of the dimples and another sister strand occupies another dimples, they came from the same original library or the same original insert from a library. And so those would be considered as two separate sequences when in fact they are the same. And so those are the four different ways that we can get artificial duplicates. And using the UMIs really can account for all of them, since UMIs are added on before PCR as well as, of course, before sequencing. Questions about duplicates before we get into trimming? Can you please repeat the sister part? Yes, absolutely. So when you have your final library, as you see on the right here, our libraries are double-stranded, right? DNA does not like to be single-stranded, and so in order to maintain the integrity of the library and to be able to store it for at least a little bit, those libraries are double-stranded. And so, of course, when we do sequencing, as you guys saw from the video, it's just one strand that's being sequenced. And so how we get our double-stranded libraries into single strand is they undergo a denaturation step before they're loaded onto the flow cell. And so just before the libraries are loaded onto the flow cell, they're heated up to separate the double-stranded libraries. And so when they're put on the flow cell, we have single strands, but of course we have single strands from both directions of our library. And so when we put those onto the flow cell, there can be a dimple that's occupied by this strand and also a dimple that's occupied by this strand. And of course, that came from the same original molecule. And so that would be a technical duplicate. That make sense? Awesome. Wait. Any other questions about duplicates before moving on? Okay, cool. Let's move on to our next step in our pipeline. So we have assessed the quality. Oh, can you explain optical? The optical duplicates. Yeah, so in here, so in an unpatterned flow cell, find a picture I want. I don't know if I have a picture of an unpatterned flow cell. There we go. Okay, so here's an unpatterned flow cell that you have right here. Unpatterned means that it doesn't have those little dimples in it. And so in an unpatterned flow cell, you essentially have a lawn of those oligonucleotides that are attached to the flow cell. And that's what our insert that we want to sequence is going to be latched onto. And so that undergoes clonal amplification. So it'll bend over, make a copy, bend over, make a copy like we saw in the video. However, if we don't have a pattern in here, we have just this lawn, what can happen is you see how these two dots here, the green and the yellow one. So what can happen is if this was one molecule from our library that latched on to this region of the unpatterned flow cell and it underwent bridge amplification, as it's doing bridge amplification super fast, it could also kind of amplify into a neighboring area. And so what would happen is that these two little regions in this example, rather than being two different ones, if it were to happen that the same molecule, when it was doing the amplification, kind of got a little carried away and started amplifying a little too far, these will look like they will have the same exact sequences, but they'll look like two separate reads to the computer that's analyzing and looking for these kind of circles that it's telling us is each a read. And so when that happens, even though that one molecule created both this amount of copies and this amount of copies, when it's read by the system, it'll think that it's two. So it'll think that it's two distinct molecules. And so it'll give us back two separate reads, but those reads will have the same exact sequence because in fact it was generated from the same molecule. Does that make sense? Awesome. Any other duplication questions? No? Okay. Hopefully I enticed some of you who are doing an internship to actually want to dive into these UMIs and remove these duplicates. I would be very happy to get that pipeline going. Okay, moving on to trimming. So we now have assessed the quality of our raw reads. We noticed some things. The quality overall looks good. We do notice these duplicates in there that we're not going to do anything with at the moment because we need a pipeline for that, which we don't have yet. We also noticed that there were some adapters that were in there. And so do you guys think that we should trim these data? Trim or filter them? Yay, nay? Put it in the chat. Yes, yes, yes. Yeah, for sure. Any reason why we wouldn't want things like adapters or poor quality reads going into our alignment step? You guys can unmute your mic or you could write it in the chat. Like, why would we want to get rid of that stuff? Not so much diminish the validity of our results. Change results a little bit. Yeah, so when we go back and, right, so we want to figure out, right now we have a whole bunch of short sequences, right? We have no idea where they came from. We don't know which gene they came from or transcript they came from. And so our goal in doing RNA-seq analysis is to figure out where those things came from. And so in order to figure out where they came from, we have to map them back to a reference genome. A reference genome being the genome build from the organism in which the samples were derived. So for our purposes, it's going to be a mouse, right? And so if we've added something or if there's something artificial in these sequences that we're trying to map back, yeah, it's going to affect our ability to assess where they came from, because if we added stuff onto it that didn't come from the actual biology, from the actual genome, then we're going to have difficulties trying to figure out where it came from. Yeah, so that's why we want to do this trimming and filtering. And also if we have reads that are low quality, we won't be confident if it's like an A, T, C, or a G. So now when we go back and map it, if we have those low quality base pairs or reads that have a lot of low quality in it, it's going to be difficult to be confident that we're mapping it back to the right location. So let's trim and filter our data. I guess we kind of went over this. So why would we want to trim or filter our data? Trimming and filtering is also known as pre-processing the data. So we want to remove those low-quality reads. We want to trim adapters. If you guys recall from looking at the quality of the base pairs from the start of the read to the end of the read, you'll remember that the start of the read is slightly lower, not too bad, but then really at the end of the read, we kind of see a falling off of the quality. So some people will actually just go ahead and trim the start and or the end of their read. We don't do that at GeneLab, but some people just do it automatically. Another thing that we want to do is remove reads that become too short. So as we're kind of trimming our reads and removing low quality reads and removing adapters, we're removing base pairs, right, from our read. And so eventually we could end up with a really short read. And so why do you think that would be a problem? Why do you think having reads that are too short would be a problem? It might match too many places. Exactly. Yes, Aylin and Hannah Lee, that is absolutely right. And so we want to make sure that we remove any reads that become just too short because it's just going to be too difficult to map them. So there are several tools that are available that do this type of trimming and filtering. And all these tools actually pretty much work the same way. Some of them use different programming languages to do what they do. Some of them have different features, but essentially they all kind of do the same thing. So here are just some common tools that are used for trimming or filtering sequencing data. So CutAdapt is one. TrimGalore, which uses CutAdapt under the hood. So it's a wrapper for CutAdapt and offers a couple more features. That's actually what we use on GeneLab. We use TrimGalore to trim. There's also Trim-O-Matic, and HTStream is another one. So there's a variety of different tools, and I put links to all of them. So if you guys are curious, you can go ahead and check out the link and learn more about these different tools. And so just like what we saw with the Unix, I like the names. So just like what we saw with the Unix, with the Unix tools and also with the R tools that Lauren went over yesterday, is that each tool or program, command, kind of using those interchangeably, will have a variety of different options. RNA sequencing tools are no different. The only thing is that some of these tools have a ton of options. And so this is where we at GeneLab worked with our analysis working group members to decide on which tool we want to use for each step of the pipeline, and then also what settings of that tool we want to use for each step of the pipeline. And again, similar to a lot of the programs, the R programs that Lauren was showing you yesterday, for a lot of these options, there are defaults. And so usually if you're unsure of what to do, the default settings are a pretty good place to start. And so I'm not going to go over all of the parameters for all of the tools just because it's incredibly overwhelming. But just to show you what it looks like, so similar to what we were seeing before, you would call them with a dash and then whatever the option is. And so just some important ones here. We want to make sure that our trimming tool knows which ASCII score, which ASCII code we're using to indicate the quality. So if you guys remember, we were using ASCII ASCII 2 plus 33 symbols to indicate our quality. So, right, because we're trimming based on quality. So they need to know what that code is to be able to know what the quality is for each base pair. So we definitely need to tell them that one. We also want to tell them what adapter sequences to look for. So these tools will come embedded with common adapters for platforms that are commonly used for sequencing. So it has built within all of those tools actually Illumina adapters, since most people sequence on Illumina instruments. And so we'll tell it to look for Illumina adapters when it's removing adapters. And then we want to give it some information about the quality. So we want to tell it what quality are we going to say this quality isn't good enough and we want to get rid of it. So we actually use the default for the quality, which is 20. So that's a Q20, which means 99% accuracy. So any base that's less than 99% accuracy will get trimmed off. And then the other thing is the length. So remember, we want to remove reads that become too short. And so we actually use the default for this too, which is 20 base pairs. And so as we're trimming based on the quality and as we're removing adapters, if the read becomes less than 20 base pairs, we'll get rid of that read because it'll be too difficult to map. And then we want to tell it if we're working with paired end or not. So the default for cut adapt is it expects it to be single end. Obviously, this is a tool is a little older. And so it has an option to let us know that it's paired. Any reason why we would need to let it know that it's paired end? Wouldn't any quality below the Phred score of 33 already get cut off with Phred 33? Why do we need the quality? So Phred 33 is just telling you what the code is. So the code is written in ASCII 2, and the base of that code from basically where it starts with how these symbols are selected is base 33. I think before some of these base 64, I want to say. Is that right, Lauren?
Yes, I think that's right.
Yeah, so there are some instruments that will give the quality scores in these ASCII 2 characters, but have the base be 64 instead of 33. And so we want to make sure that the trimming tool knows what this code is and what the base of this code is. So that's what we tell it for Phred 33. So, and then it's going to read and interpret that, and then it'll read and interpret that. And as it's reading these ASCII 2 characters, it's going to look for any characters that are below this Q score of 20. So Q of 20 being a 99% accuracy. And basically it's going to read and interpret this ASCII character, the trimming tool is. And if it sees an ASCII character that represents anything below a Q score of 20, it'll get rid of that base. Okay, and then we were just going through, anybody know why we would want to tell it, why it matters to tell it about paired end when we're doing our trimming? Guys, remember what I told you about the quality of forward and reverse reads? Are they the same quality, or does one have better quality than the other? Forward is better, yeah. And so what can happen is so when you're trimming, you're going to be trimming both all your reads, right? So you're going to be trimming both your forward reads and your reverse reads. But of course, your forward and reverse reads are sequences of the same molecule, right? Just in two different directions. And so it's important, if you're telling your tools that you have paired end reads, that it actually has a pair for every single read that it has, right? So that each read has indeed a forward direction and a reverse direction. So now if you're trimming these kind of individually though, so you're trimming your forward reads, you're trimming your reverse reads, what's the likelihood that you're going to end up having to remove more reverse reads than forward reads, or vice versa, because as you're trimming, the length gets to be too small? Do you anticipate removing more likely to remove reverse reads? Yes, absolutely. And so what happens at that point is that if we don't tell it that it's paired, then it'll keep anything that passes your length cutoff, right? And so what can happen is if it removed more of the reverse reads than the forward reads, you might be left with forward reads that don't have a partner, that doesn't have a mate. And so what this paired option does is it says, hey, as you're going through and removing reads, at the end, let's make sure that if we removed a reverse read, we also remove that respective forward read. So that way we always maintain that there's a forward and a reverse read for every molecule we have. Make sense? Awesome. Okay. So what happens after we trim our data? So we're going to be doing this. I promise, I know we're taking a little longer, but I'm really happy that we're diving into the concepts here. They're very important, and we're going to see all of this as we process the data ourselves. And so just taking a look real quick, I'm not going to go through all of the quality metrics for the trim data, but remember, right after we trim, we do another quality assessment. So we want to check the quality of our data after trimming to see if we did indeed improve our quality from our raw data. And so I'm just going to do some comparison. This is still that same sample, except this is that sample after we trimmed with those settings that we just talked about with Trim Galore. And so just for comparison's sake, I have the trimmed or filter data on the bottom, and just to remind you, I have the raw data on the top. And so we're going to ask some questions. So how many reads were removed during pre-processing? You guys go ahead and put your answers in the chat. So this is the data we started with. This is the data we have after filtering for this particular sample. Around 150K years. That is absolutely right. So how do we know how many reads were removed? Well, we can look at the FastQC analysis. There's other ways to do this too. We can look at the FastQC analysis though, which is one way, and we look at the FastQC of the raw data. So we started with 100,000 or 100,600,000 to 211 total reads for this sample. And then after trimming, we have 100,000 or 100,453,545 reads. And so to know how many reads we got rid of, you would just do the math of taking the number of raw reads, subtracting it from the number of reads we're left with after trimming, and you get about just under 150,000 reads. What's the sample read length after trimming? Yeah, 20 to 50. Any guesses why that is? Why 20? Why is 20 the smallest? You can put it in the chat or unmute your mic. Yeah, well, it's actually Trim Galore we used, but yes, you're right. So we use Trim Galore, which is cut it up under the hood, and we use the default limit, which is 20, right? So that's going to be the smallest we have. And then, of course, we know that if nothing was trimmed, then we would be left with 150. So we do see some reads in which nothing was trimmed away. So that's why we have that range now. So let's take a look at the quality. So the per base quality here. So what do you notice? How has this per base sequence quality changed after trimming? Look better, look worse? Yeah, it looks better. So we're no longer seeing this kind of dip into the below Q20 score, right? And then just to get you guys thinking, what would happen to the quality if we increase the Phred score cutoff to something above Q20? Yeah, we would see, yeah, even tighter in the green area. Yeah, so we'd have even better quality if we increase that score. But a 99% accuracy is pretty damn good if you ask me, so I think we're okay. Looking at the adapter content, what happened to the adapter content after trimming? Yeah, no more adapters. So we successfully removed our adapters. Awesome. And so that's kind of all the comparisons I wanted to show you guys between trimmed and raw filtered QC. I think we are officially ready to get out of here and let's do some work. So I want everyone before the break, I know we have a break scheduled for 10:40. I want to stay true to that. But before the break, I want everyone to make sure that they can log on to their to San Jose State VPN and log on to log on to the Jupyter Hub. I'll put that Jupyter Hub link in the chat again. So before you head off to the break, let me know if you do not have access to your Jupyter Hub. If you do have access to your Jupyter Hub, you can take off on your break a little early. I'll hang out for another couple minutes before I take a break. So please write in the chat. Everyone, before you leave, pull up your Jupyter Hub. Make sure you have access to it. If you're having issues with access, message me in the chat. Okay, if no one's having issues, go ahead and enjoy your break and be back here by 11 a.m. and we'll get started right away on analyzing GeneLab RNA sequencing data. I'm going to hang out for another minute or two in case you're having trouble with Jupyter Hub. So message me in the chat, and I'll be back a few minutes early too. I'm going to stop my recording.